It's a real pleasure to be here today representing PlusVital and to tell you about the recent developments in our genomics research and in our development of novel supplements. I was here this time 8 years ago, shortly after we launched the speed gene test, and a lot has changed in that very short space of time in terms of the developments of the technologies and how we can apply those technologies and the knowledge that we have about how these can be used in practice. When you think about it, 8 years is a very short space of time. When you consider that for hundreds of years, breeders have been doing very similar things in terms of breeding decisions and selection decisions. But one thing that breeders have always done is that they have recognized genetics. They've recognized that genetics is fundamental to making the horse the individual that it is, because they have always recognized and considered pedigrees. Pedigrees attempt to reflect the genes that have been passed on by ancestors in that pedigree to that individual. Now, it's not that simple. I'm going to show you why it's not and how, using genetics, you can actually drill down through the pedigree into the actual DNA makeup of a horse and how that can be informative. As I mentioned, over the last 10 years, there have been significant advances in the science of genomics for the horse, but not only for the horse, in all mammalian species. The horse genome was sequenced in 2010. That means that we now know the 3 billion units of the genetic code that spell out the instructions to make an individual horse. And it's our job to try to decipher that. Not only do we have that knowledge, but we also have the tools, the laboratory tools and the computational tools to interpret that information. PlusVital has been in the equine business for 35 years. PlusVital is an equine supplements company that's been providing nutritional solutions to the global thoroughbred industry and other equine industries internationally, as I said, for 35 years. Econom, which was my company that I started in 2009 in collaboration with Jim Bolger, was developed out of a university research program that I was involved in, and we launched the speed gene test in January of 2010. Econom and PlusVital came together in 2015 to provide better solutions for the horse industries. We have 15 years of research in the area of equine genomics. We are considered the global leader in this field. We've had significant levels of funding in our research, and we have a very highly skilled team of researchers, many at PhD level. Not only that, we have over the last 8 to 10 years accumulated a huge resource of DNA samples. We have 20,000 DNA samples that we can use in our research to try to identify genetic profiles that differ between individuals. Not only that, instead of looking at single genes at a time, we can now look across the entire genome, and at a single moment in time, we can look at 70,000 units of DNA in an individual horse at a time. So it's extremely powerful. And over the last 8 to 10 years, there have been giant steps made in this field. Well, genetics is a huge part of what makes an individual, but there's also another large part that makes an individual, and that is the environment. It's not a battle between nature versus nurture. What we really should be considering when we're considering genetics is that genetics sits alongside the environment and the management of the horse, and together nature and nurture makes the individual. So what we are trying to do in understanding the genetic contributions to performance traits or traits of importance in the horse is capture just that genetic part of the trait, and with that knowledge we can then maximize the opportunity of the environment. As I mentioned, there are 30 billion units of DNA that we now can understand. These are the sequences of the letter of the DNA code, the G, A, T, and C, that some of you might be familiar with. There are about 20,000 genes in the horse genome, and each of these genes encodes a protein or spells out the instructions to the cell to make a particular protein. There are variations in this in terms of when those genes are turned on or turned off, and this is a phenomenon known as gene expression. And the environment can influence this as well. Well, what we are looking at in looking at our DNA profiles is actually just the inherited genetic differences that have been passed down from parents to the progeny. Now, in the inheritance of these traits, very few of them are what we refer to as simple traits, whereby one gene is responsible for the entirety of the trait, that it's entirely genetically coded by or influenced by a single gene. An example of a simple trait would be the grey coat color in horses. That is determined by a single gene. A grey horse must have at least one grey parent, and it is either grey or not grey. There's no in between, and that's caused by a single gene. That's very, very unusual. Most traits are, particularly in the thoroughbred, are complex traits or polygenic traits, whereby many, many genes contribute to the overall phenotype, or what we can actually see. And not only do these many genes contribute, they all interact in different ways with one another. And to complicate the issue even further, the environment can interact with these genes. What's important is a term that we refer to as heritability, which is the relative contribution of genetics to the trait. So in some cases, a trait will be entirely heritable. So that's an example of, say, the grey gene. And in other cases, there'll be no genetic influence to a trait. I asked some students recently if they could give me some examples of what might be something that is only environmental, that doesn't have a genetic influence, and somebody came up with the idea of accent. So my mother is Australian, but you probably wouldn't know that from my accent. So accent is not genetically determined. So the heritability of that would be 0. So we're looking for traits that do have a genetic contribution. And if we know the genetic contribution to a trait, then we can maximize the environment for that individual. So genetics and the environment or management go hand in hand together. The reason that foals are not identical or clones of their parents, and why full siblings are not identical to one another, is that at every generation of the gametes, the egg and the sperm cells, it's a complete lottery of the package of genetic information that gets passed down. It's also a complete lottery of what genes come together at the fusion of the egg and the sperm cells at the point of fertilization. All horses have 32 pairs of chromosomes, and that's what we refer to here as the N. N is 32. So all horses have 2 copies of the 32 pairs, one inherited from the sire, one inherited from the dam. Now they can't pass on all of those, both of those pairs. They can only pass on one set, because otherwise the foal would have 64 chromosomes, and so on, and it would be duplicated. So at the generation of the gametes, the egg and the sperm cells, these packages of chromosomes have to be split in 2, so that only one set of the 32 chromosomes is passed on. That's why each individual gets only 50% of their DNA from their parents, from each parent. What happens then at this splitting of the chromosomes into packages is that there's another complication. The genes actually physically get mixed up. This is something known as recombination, which means that the grandparental genes all get mixed up and shared before they get packaged into these chromosomal units, which are the egg cells or the sperm cells. Then at fertilization, it's completely random chance of which of these packages come together. Because there are 32 possible combinations, this means that there are actually 4 billion individual combinations that can arise. So Full siblings have a 1 in 4 billion chance of being identical genetically. Essentially, it's impossible for full siblings to be identical. What this means is that at every single generation of a new zygote or a new embryo, there's a shuffling of the deck of cards, and it's completely random what comes together. So you might look at your pedigree and try to best guess what's going to come about, but it is completely random. What we can do is, and actually what breeders do in pedigrees, is they try to reduce that randomness by choosing horses that they think might suit or might have similar genes that are inherited from those ancestors that will come together in the right package. What we can now do is we can look at the package that is there and identify the genes that have actually been inherited. The best example of this, and the best known example of looking at the DNA of a horse, is what we refer to as the speed gene. There are over 35 scientific publications in peer-reviewed journals now that support our original findings that we published in 2010. Some of you may be familiar with the terminology that we use. If a horse has inherited a C-type chromosome from both the sire and the dam, then we refer to them as CC horses. If they inherit a C from either the sire or the dam and a T from the other parent, then they're what we refer to as genetically as CT, and then the TTs are if they've inherited a T-type chromosome from both parents. The CCs we have shown in multiple studies are the sprinter types. So these are the individuals that prefer short distance sprint type racing, compared to the CTs that are the middle distance horses, and the TTs are the horses that prefer and do better over longer distances. There's a reason for this. We now know what that reason is, and it's because this, the mutation is actually linked to the C version. The mutation changes the production of the myostatin protein. The myostatin is the gene where the mutation is, and it changes the production of the protein. Myostatin acts as a brake in muscle development. So when the brake is on, when myostatin is there, the brake is on and muscle doesn't grow as quickly. When the mutation is there, that protein is not produced, the brake comes off and muscle grows. And the best example of these prior to the horse is what we see in Belgian blue bulls that have this double muscling phenotype. They look like that because they have 2 copies of mutation in their myostatin gene, one inherited from their mother and one inherited from their father. The dog in the middle is a whippet, believe it or not. We refer to those dogs as bully whippets, and they look like that because they have 2 copies of a mutation in their myostatin gene. That's detrimental to performance because they have too much muscle bulk and they can't run as fast. But dogs that have 1 copy inherited from either parent are actually better racing dogs than dogs that have no copy. So we've also shown this in thoroughbreds, that the change in the protein production that's caused by the mutation actually alters muscle growth. And CC horses have significantly more muscle mass as 2-year-olds than TT horses. 7% more muscle mass as 2-year-olds than TT horses. The other thing that this gene does is that it alters the proportion of muscle fiber types. So we and other researchers have shown that CC horses have significantly more of the type 2B or type 2X muscle fiber types. These are the fast glycolytic fiber types that are required for short bursts of speed, which makes sense in terms of the requirements for sprinting. Because of these factors, because of these functional changes in the muscle, this has an effect on how these horses perform on the race course. We now, over the last 8 years, we've made this discovery that CCs are better suited to short distance and TTs to long. But more recently, we've put this in the context of how they actually perform on the race course in terms of the value and the prize money that they earn on the race course at different distances. And it all differs in different geographic regions. In Australia, it appears that the race pattern is strongly biased towards the shorter distances. So this is data specifically for Australia from a sample size of over 100,000 races, we find that the majority of races are competed at the 1,000 to 1,200 metre range. And this is reflected in the prize money, where the majority of prize money is earned in the 1,000, 1,200 and 1,400 metre range. So there's a huge bias towards sprinting or speed in the Australian population, and that's actually reflected in the genetics of the population. So in Australia and New Zealand, of the horses that we have identified and sampled, 48% of the population are these CC types. That's quite different from Europe. In Europe, it's now at about 35%. It used to be about 27%. It's increased in Europe in recent years. So the majority or genotype, or the biggest proportion of horses, are CCs in Australia. And you might think, well, that makes sense because of the distribution of racing. But if you think about it, 48% are CCs, 52% are not. And it's these individuals that are not are the ones that are most difficult to try to figure out and place in most suitable races. The speed gene test, or this information, the CC, CT and TT, can be applied to make decisions at various different stages of the life cycle of the horse. And I'm going to go through these with particular emphasis on horses in training. CCs, we now know in Australia, perform optimally over sprint race distances, and as 2-year-olds perform best over the 1,000 to 1,200 metre mark. We know in 3-year-old races they can race on over slightly longer distances, but they do have a limit. 88% of CC horses have a best race distance of up to and including 1,600 metres or 8 furlongs. If we look at 2-year-old performance and 3-year-old performance, these values actually get a bit tighter. Not only does it influence how they race, or the type of racing that they're suited to, but it also influences their precocity. We know that 50% of CC horses have their first race at least 1 month sooner than the average CT horse, and at least 5 months earlier than the average TT horse. So they're the more precocious types. They also look better in the sales ring. These CC horses are the ones where the brake comes off quicker in the muscle, so the muscle grows. They are they're the ones that also have more muscle mass than as 2-year-olds. The CT horses are more versatile in terms of the distances that they are best suited to. As 2-year-olds, you'll see the data now, they are best, we recommend that they're best targeted at 1,200 metre races and not the 1,000 and 1,100 metre races. And then as 3-year-olds over the middle distances. The TT horses are at the complete opposite end of the distance spectrum, and the majority of TT horses have a preference for racing over 1,600 metres. If we tighten this up to 10 furlongs and above, it's about 80 or 90% of horses, of TT horses, have a preference for those really longer distances. During the 2-year-old season, we know that there's a restriction on race distances. So we applied our sample size of 1,600 horses that we had tested for the speed gene test and looked at how they performed at the different distances as 2-year-olds. The top chart shows the percentage of runners by distance according to their speed gene type. The CCs are the purple, the CTs are the blue, and the TTs are the brown. So what this top chart says is that 61% of horses that ran in a 1,000 metre race as a 2-year-old were CCs. But they actually won 83% of the prize money. So they were overperforming at the 5 furlong distance. The CTs, on the other hand, while they 32% of runners in these 1,000 metre races were CTs, they only actually won 17% of the prize money at those distances, at that distance. Similarly, for the 1,100 metre races, the CCs were by far outperforming the other types in terms of the achievement of prize money. And it's not until the 1200 metre races become available or apparent for the 2-year-olds that the CTs start to come into their own. So we recommend targeting racing for CTs as 2-year-olds over 1200 metre races. Now, what may be happening here, and I recognise in Australia that oftentimes horses are tried over the 5 furlong, or the 1000 metre distances and then progress on. That's what trainers like to do. They give their horses an education and they try to figure them out as they go along. And this might be useful in the absence of this information, but with this information, it's not as necessary. It might be useful for the education, but not necessary to figure them out. So we have very, very strong evidence now that this information is valuable in terms of placing horses in the races that best suit their genetic type. What's interesting here also is that the TTs, 6% of horses that raced in 1000 metre races were TTs. These are the longer distance types. And they won no prize money as 2-year-olds in this sample set. Now you might say, well, 6%, that's not very much, but actually that's 6 out of 100 horses. That's 3 out of 50 horses. I'm not sure that anybody can afford to take a chance with 3 of their 50 horses that they're not actually earning at this distance. The other problem with this is that TT horses require, because they're stayers, they require a greater workload to build up their aerobic exercise potential, and increases in workload are associated with increased injury risk. Some data that I heard the week before last is that over 50% of Australian horses in training actually exceed the risk point for the amount of cumulative workload that they do in training. So this is actually putting these TT horses at increased risk of injury by racing them in these and training them and racing them for these distances at an early stage. As 3-year-olds, the pattern is quite similar, but what was quite surprising to me is that at the 1000 metre distance, you still have 36% of runners are CTs, and they're underperforming at this distance. Now that to me is not trainers putting them in to educate them or train them. It appears that they're targeting these horses for those races, and they're underperforming. The CCs are overperforming at these shorter distances. And then you can see the TTs in the brown are coming into their own over the longer distances. Sometimes prize money can skew these figures because a large prize fund can actually skew the differences. So what we look at here is strike rate. Strike rate is wins to run ratio, and the percentage winners for the different genetic types. And it's consistent. The pattern is absolutely consistent. The CCs are outperforming at the short distances and the TTs at the longer distances. For every horse that we do a genetic test, we collect a blood sample and we return the results in a report format like this. These are a new report format with the genetic type of the horse, and then all of this information for interpretation. So it should be straightforward to interpret this data and where to place your horses for the best outcome on the race course. It's also useful, of course, for breeding decisions. I mean, some people will say, well, you know, we can tell this from the pedigree, or we can tell this by, you know, looking at the ancestors. It's very much a staying pedigree. It's very much a speed pedigree. But if you have a CT mare, and you take her to a CT stallion, the same CT stallion, 4 years in a row, you could, in fact, get very different genetic types in the foals. So if the mare passes on, the sire and the dam will only pass on one of their copies of the speed gene at each time, as I've said. So if the mare passes on her C and the stallion passes on his C, it's a CC. 50% of the time that mating will produce a CT, and 25% of the time that mating will produce a TT foal. So these full siblings could be completely different genetically. They will have exactly the same pedigree page, but they can be very different genetically. We've already seen the CCs are going to perform better and earn more over the shorter distance compared to the TTs. And this is often where owners and trainers will get it wrong, where they'll have maybe a CC horse in 1 year that's very sharp and early maturing and fast. Their full brother comes in the following year. It may be a TT, and that may not be recognized, and instead it may be considered that that individual is just slow or no good. That's not necessarily going to be the case. It's just that they need more time, and they need to be prepared and competed over the longer distances. So the pedigree does not tell you everything. We do know, though, if you have a CC mare or a CC stallion, they can only produce CC and CT types. They will never produce a TT type because they don't have the T to pass on. A CT mare can produce all different types, and that's the same for CT stallions. That's why you have some stallions that are very versatile in the types of foals that they produce. They produce very different types. And similarly, the TT horses will never produce CCs. So you can use this information to manage your matings. If you have a mare and you want to ensure that you're only producing CC types, then you can choose a stallion that best suits her for the preferred outcome. The speed gene has also been used in the sales ring. In Australia, we collected samples before yearling sales in 2004. And we didn't know the genetic types at the time. We subsequently went back and looked at how much they achieved in the sales ring, and then how they performed later on the race course. The CCs, 55% of the horses that we sampled that went through the sales ring were CCs, and they achieved approximately $30,000 more in the sales ring than CTs, and significantly more again than the average TT. And this is because the removal of the brake of the myostatin gene that creates muscle mass is actually stimulated by exercise. So when yearlings are brought in for their preparation for sale, even the exercise stimulus on the walker or walking the horse will actually accelerate the removal of that brake and result in greater muscle mass. So those CC horses tend to be the ones that are actually looking more precocious and looking better in the sales ring. Not only that, then they went on as 2-year-olds to have significantly higher earnings on the race course and higher strike rate. So 42% win or place per start versus 24% and 19%. So what we, all in all, to summarize the speed gene test, we have identified a single gene. It has an extremely strong scientific association with muscle traits and race distance. The genetic test that we have developed on the basis of this scientific information is an extremely accurate predictor of race distance. And using the speed gene test in, I suppose, the most appropriate way can actually lead to an economic benefit for the owner on the racetrack and in the breeding shed. The speed gene is just one gene. It appears to have an unusual singular effect on the distance trait. But there are many other genes that contribute to an elite racehorse or to the various other traits that are important for success of a horse. So we have developed a range of other tests, including what we refer to as the elite performance test, race-done-race test, which is the chance of a horse having at least one race course start as a 2-year-old or a 3-year-old, and preference for racing on different surfaces, and particularly in the US, where there's a major selection for dirt horses versus turf horses in North America. These tests are based on assessing thousands of genes at a single time. In the very beginning, I mentioned that we can now look at 70,000 units of DNA at a single time that are representing these thousands of genes that are contributing to these traits. What we do then is we generate genetic profiles based on these markers. And what we're trying to do is we're trying to identify the extremes of a normal distribution. So all horses have different versions of these genes, but the horses at the top end will have a certain package, and the horses at the bottom end will have a certain package of them. So this is what we're trying to do. We're trying to profile the top end versus the bottom end. And this allows us then to make predictions by profiling the top end and the bottom end of known horses. We can take an unknown and see where it best matches. One of our tests, we call the race-to-unrace test, is actually influenced by two main genes, one called NTM and one called PRCP. Now, race-to-unrace we refer to as the chance of a horse having at least one race course start as a 2-year-old or a 3-year-old. And we found that the major gene contributing to this is the same gene that influences the motivation of a mouse to voluntarily exercise on a wheel, a running wheel. Some of these mice that have a mutation in this gene, they're the mice that will spend up to 20 hours a day running on one of these wheels in their cage. And then there are other mice that won't go onto the wheel unless they're actually prodded with an electric shock. So it appears that this gene is influencing the motivation to exercise, and it's exactly the same gene that we find in the difference between horses that have a race course start versus horses that do not have a race course start at 2 or 3. So we think that this is a gene that's influencing the motivation of the horse to exercise. We all know that there are some horses that are really keen and eager to go out. They're anticipating exercise in the morning, and they're wanting to go out and do their work. And there are other horses that trainers will describe as having a poor attitude, can't really be bothered, aren't up for the work. Now, this isn't to write these horses off, the horses that have low genetic potential for having a race course start. It means that you may be able to use this information to manage the environment better for those individuals, to try to stimulate them or motivate them in their exercise, having this knowledge in hand. Horses that we've profiled as being high potential for a race course start are 2.6 times more likely to actually have a race course start. So when we looked at this, is actually the results from the yearlings that we collected at the yearling sales. And again, we didn't know this information before they raced. It was only after they had raced. The horses that we tested that were high potential for a race course start, of those, 44% actually did have a race course start. The horses that we predicted to have a low potential for a race course start, of those, only 17% actually had a race course start. So this is a very accurate predictor of the chance of a horse having a race course start based on a genetic profile. This also translates into those individuals that actually did have a race course start. Those horses that were lower potential had significantly lower earnings. So even though they did have a race course start, they weren't competing in high level races at all. Another multifactorial trait is the preference for racing on dirt or turf surfaces. And in fact, this actually does translate into the preference of horses for different ground conditions, firm versus heavy ground conditions on turf. And this test has a very high accuracy, where we can predict horses that are best suited to racing on dirt, and horses that are best suited to racing on turf. And funny enough, when we look in the European population, the majority of horses, as you would expect, are predicted to be turf horses. In the US, it's about 50-50. The genes underlying this appear to be genes that are controlling the transition of gait. These are signaling genes that are involved in the neurotransmitters that may be functioning either in the brain or may be functioning in the musculoskeletal junction, in the signaling from the nervous system to the muscle in how to respond. We know actually studies that have been done here in Australia have shown that sprinters tend to have a higher frequency of stride pattern compared to stayers, and this appears to be the same case with dirt performers versus turf performers. So this is a very accurate test that we're now using mostly in the States. We have an ongoing research program where we are using our 20,000 DNA samples and looking at 70,000 genetic markers in those samples and looking at a range of different traits, partitioning them into the extremes, looking for the for example, the response to training, the high responders to training versus low responders to training, profiling them so then we can make a prediction. You'll notice here there are a number of inherited disorders, EIPH or bleeding, RLN, recurrent laryngeal neuropathy or roaring. These all have been shown to have an inherited contribution to the trait, and we're now starting to be able to identify the genes that may be used to screen horses for a higher risk of developing these diseases, many of which are progressive in nature and are also influenced by the environment. So if they're influenced by the environment, if you know the genetic risk, you can modify the environment to try to minimize that risk of actually occurring. So using these new genetic data, you can optimize training and racing programs that will, and we have shown, we have evidence now that using the information can actually increase your return on your investment for the horse. You can use it to achieve higher sales prices if you're targeting the production of CC horses that are going to show better in the sales ring. You can run a more efficient breeding program if you know the genetics of your mare and if you know the genetics of the stallions. That's beneficial. In Australia we have quite a few farms now that are advertising the genetics types of their stallions, and more are coming on board. Widden, for instance, has the genetics, the speed gene types of all of their stallions, and they advertise those. They're all on their website. And we have other farms now that are also providing this information for their stallions. We've tested over 300 stallions worldwide for a range of farms. So genetics will continue to work on identifying genes that can predict performance traits, and there are many different performance traits that we can look at, but also for looking at risk of inheriting important and debilitating health traits, as I've mentioned. A new area that we have got into from the genetics field is the science of nutrigenomics. This is whereby you would identify the nutritional requirements of an individual based on their genetics. We all know that you can feed the same horses exactly the same. You can feed a bunch of horses exactly the same thing, but they will respond, some of them will respond differently to that feeding regime or nutritional regime, and that's likely because of their underlying genetic differences. So last year in Europe and just this year in Australia, we launched the world's first nutrigenomic supplement for horses. So this is a scientifically based research supplement that is targeting horses with a specific genetic type. Nutrigenomics is the science of the interaction between nutrition and genetic differences between individuals. And the idea is to optimize the nutritional regime with respect to an individual's genotype or genome. And what we found is that horses with the different speed gene types actually have different requirements for a nutritional element called CoQ10 or coenzyme Q10. And therefore we developed a product that we call EnerGene Q10. The reason that this there are different requirements for CoQ10 in the CC, CT and TT horses is that we know that sprinters and stayers have very different metabolic and physiological requirements for the different exercise that they undertake. Energy is produced predominantly through two main pathways: the aerobic pathway, which uses oxygen and is very efficient in producing very high levels of energy in the form of ATP, versus anaerobic production of energy, which doesn't use oxygen, uses other substrates to produce energy in the form of ATP, but it's not as efficient. Horses that are trained and competing over longer distances tend to depend more on oxidative or aerobic production of energy, whereas sprinters tend to rely more on anaerobic production of energy. And that's because it takes the lag time that takes a considerable amount of time for oxygen to be taken into the horse and into the lungs and then delivered to the muscle. And it's estimated somewhere around 50 seconds. So by that point, most sprint races are close to being over by the time oxygen can actually be delivered to the muscle to be used in aerobic production of energy. So sprinters tend to rely heavily on anaerobic. For aerobic production of energy, oxygen must go through this cycle. It's called the electron transport chain, or the Krebs cycle, the electron transport chain within the Krebs cycle in the mitochondria to produce ATP. Oxygen, to be converted to ATP, requires this coenzyme Q10. It's required in order to move the oxygen along this transport chain to be converted into energy. Any deficiency in CoQ10 in the muscle cell there will be a required move from aerobic production of energy to anaerobic production of energy. And this causes fatigue, because the byproduct of anaerobic metabolism is lactic acid. And lactic acid buildup in the muscle is what actually causes fatigue. So you want to try to prolong the aerobic energy production as long as you can, and the way to do that is to make sure that you have sufficient CoQ10 supplies in the muscle. The reason that it's different between CCs and CTs and TTs is that the different types have different proportions of the different fiber types. I already mentioned at the very beginning that CC horses have significantly more of the type 2X fibers. These are the fast glycolytic fiber types, so they're anaerobic fiber types. And then versus the oxidative fiber types, the type 1 and the type 2A fibers are the oxidative fiber types. TT horses also have more mitochondria than CC horses, so you would expect the mitochondria are the energy producing parts of the cell where this conversion of oxygen to energy takes place. TT horses naturally have more mitochondria because they have more of the type 1 and type 2A fibers. But we found that they have less of this CoQ10. So you can see here CoQ10 is the cog that is required to convert oxygen into energy. We found that TT horses naturally produce less of this CoQ10. So even though they have more mitochondria, they have less efficient mitochondria, less efficient in converting oxygen into energy. What we found is that when we supplemented in the lab, so this is in vitro in petri dishes essentially, when we added CoQ10 into muscle cells that were CC, CT and TT muscle cells, we were able to restore the levels back to the average levels for CCs and CTs. In a field trial, we also looked to see if we could do this in vivo in live horses. So we had access to 19 thoroughbreds that were untrained. They were all kept in the same farm under the same management conditions at pasture. And we supplemented them, or orally supplemented them, with CoQ10 by syringe feeding them every day for 9 weeks. So that's 19 horses every day for 9 weeks with CoQ10. And we took muscle biopsy samples from these horses before the supplementation period and after the supplementation period. And we found that the CoQ10 levels increased by 40% in the muscle after supplementation. There's two really important things that this tells us. The first thing is that the CoQ10 that we have in our EnerGene Q10 is bioavailable. There are very few supplement products that actually show that the supplement gets to the site of action where it's required. It may be a case of in one end and out the other, unless it's actually demonstrated that it gets to the site where it's working. So we now know that the EnerGene Q10 gets to the muscle where it's needed, and that supplementing with CoQ10 can significantly increase the levels of CoQ10. This should then allow for more efficient energy production, because it'll allow a more sustained use of aerobic production of energy. It will delay fatigue by prolonging aerobic capabilities. As I said, you want to prevent the switch to anaerobic metabolism, because that's where you're going to get the lactic acid buildup and therefore fatigue. So it will delay fatigue by increasing the amount of time that horses can exercise aerobically. Another really important point is that horses that are supplemented with CoQ10, we expect to have faster recovery between exercise. That's because during rest periods between exercise bouts, so I mean work days or high intensity exercise, the main point of that rest period is to replenish glycogen stores, and during that process they need to use energy in some way. So the major energy pathways are aerobic. So if you can actually fuel their aerobic pathways, they will actually recover quicker from high intensity exercise and should be able to exercise more. While we do think that this product will have greater effects in TTs because they are more dependent on aerobic energy production, we do believe that this will also be beneficial for CCs in the training regime, because they should be able to recover from their exercise more quickly. So this product is now on the market here in Australia. Kelvin from Omicron is here, and he's our supplier in Australia. Hopefully all of you have signed up for the draw at the door for a tub of EnerGene Q10. If you haven't, please do on your way out. We're also offering a speed gene test as well to the lucky winner. But it's definitely worth a try. We have over 40 trainers here in Australia that are using the product. Many of the Melbourne Cup runners were on this product this year, we're delighted to say. Thank you so much all for coming.